But we're going to start our first presentation will be by a shared fellowship. Dr. Agbash worked in his lab with Alphaba and Qwynton Johnson, title of which is Profile of Post-Translational Modifications of TDP43 in Neurodegenerative Diseases, a blood-based biomarker development. Take it away.
Hi, my name is student Dr. Alphaba, and I work with my colleague, student Dr. Johnson in Dr. Agbash's lab. The title of our presentation is the profile of post-translational modifications of TDP43 in neurodegenerative diseases, a blood-based biomarker development. Our hypothesis is that post-translational modifications of TDP43 can be assessed in platelets of blood obtained from patients with Alzheimer's and with ALS. Without getting into too much detail, TDP43 is a binding protein that regulates mRNA splicing as well as gene transcription. And this protein undergoes many post-translational modifications that leads us to accumulate in the platelet of people with neurodegenerative diseases. Therefore, our aim is to determine the detectability of these post-translational modifications of TDP43, and then we'll compare the most prominent of those modifications of both diseases to the age-matched healthy subject in order to create a repertoire that can lead to an easy diagnosis. Over the summer, our project consisted mostly of doing Western blot analysis, but we had also the opportunity to do a high-performance immunoprecipitation, HPIP. This procedure allows us to isolate our protein of interest, in this case, the TDP43, from a pool of protein that are normally found in the platelet sample in a matter of a couple of hours rather than days. The way that it works is that we install TDP43 antibodies in the tip of our pipette that you guys can see in the picture. And from there, we run our sample through it, and the antibodies will bind to the TDP43 and wash out the rest of the protein. And then we'll obtain the concentrated TDP43 and then do a Western blot analysis on it. Our panel consisted of ubiquination, sumolination, acetylation, and cysteine oxidation. We also looked at phosphorylation because phosphorylated TDP43 derivative have been detectable in platelet lysate samples. I'll now pass down to my colleague, student Dr. Johnson, who will talk about the results.
Hello, everyone. My name is student Dr. Johnson. I'll be discussing the results of our research this summer. And so here we have the Western blot analysis. Using LICOR imaging software, we can measure the signal intensity from each antibody. And the higher the intensity means that there's more protein present. And so on the left top image there, that is platelet cytosol from patients with ALS. And on the right, you have quote unquote healthy platelets, so patients without ALS. And this graph in the center shows that for SUMO1, SUMO2, 3, ubiquitin and cysteine, the signal was more intense in control subjects, and the lysine was the only one that showed brighter intensity in ALS positive platelet sample. And this is the same thing, but we're talking about Alzheimer's disease now. And there's a bit of a difference between ALS and AD, as you can see, where SUMO1, SUMO2, 3, and cysteine showed higher signal intensity in Alzheimer's disease platelet sample, whereas the control showed higher signal intensity in ubiquitin and lysine. Next slide. And so some conclusions can be made here, but frankly speaking, we need a lot more experiments to make a conclusion. And so currently we are working on more analysis, as well as looking at more post-translational modifications, including glycosylation studies. Yes. As a quick thank you to Dr. Agbash for allowing us to work in his lab, Adina for all the support and guidance, KCU and the SSRF program for allowing us to do research, and the rest of our team Q for help and support. And now we will have a Q&A session. Yeah, Dr. Zaidi.
Well, thank you very much for an excellent presentation. I have a quick question for you too. In your results, as far as I understand, I saw the Western blots for the disease sample on the left and the controls on the right. So my question was, did you run those blots separately, or did you have the controls and the disease within the same gel? Because there's gel to gel variation, and it's kind of difficult to compare two separate blots. I'd rather have see the controls and the disease within one gel to avoid those errors.
Right, thank you, and that's a great question. We did use different gels for the pathologic samples compared to the healthy samples.
So I would suggest you to do the control and the disease within one gel to avoid variability. Because every time you develop a Western blot, the intensity is going to be different. There's just little human errors in loading and across gel variations, and I'm thinking you probably will get very small changes. So you want to make sure that you avoid all kinds of errors and have the sample within one gel if possible.
Okay, that's great advice. Thank you.
Thank you very much, Alpha and Qwynton. Excellent job.